Good morning. Guten Morgen. Bonjour. My name is Jan Leach, and I serve as the president for the International Society of Plant Pathology. It is my great pleasure to welcome you to the International Congress of Plant Pathology 2023. This wordle was constructed using the welcome word from all the languages of 100 different countries, and those countries are the countries that are represented in this room. Now, I'm not going to stand here and say welcome in every single language, but I want you to work with me to teach everyone how to say welcome in your language. So on the count of 3, shout out welcome in your native tongue. 1, 2, 3. Oh, you guys are good. Great. Even after that big party last night, you guys are great. So it's so wonderful to see you here in person in Lyon. I know many of you traveled for many, many miles to get here from great distances, but I want to especially give a shout out to our environmentally minded Zero bikers who rode their bikes from Montpellier, Avignon, all the way to Lyon, and they were speaking to reducing the carbon footprint that we daily impart on our environment. Now, in recognition of the many kilometers that these wonderful bikers rode, we're offering you the opportunity to donate to the ISPP Resiliency Fund using the barcode shown at the bottom. The International Society of Plant Pathology is an umbrella organization, and we represent over 63 different societies. And by default, if you're a member of any of those societies, you are a member of the International Society of Plant Pathology. So collectively, we estimate that we represent, and you are part of, a community of over 26,000 plant pathologists or plant health specialists internationally. One of the things that we do is our charge is to convene and to bring our members together to help connect the community of 26,000 plant pathologists around the world globally. So we do this in 2 different ways, in several ways, but one way is this International Congress. We convene meetings once every 5 years. But the other way is that we have built a network or a website which has a directory, a worldwide directory of plant pathologists. And in order to help you connect better with your international colleagues, we invite you to introduce your information into this website at the barcode. Everything's barcodes now, right? So the mission of the International Society of Plant Pathology is to promote worldwide plant health and food security. Now, given the this mission statement, this is a particularly urgent matter in this day and age. We know that there are huge needs out in the world, and this report from the Global Report on Food Crises, which was issued this year, points out that over 250 million people are acutely food insecure worldwide. This number is the highest. It's not going down. It's the highest since this report was initiated about 7 years ago. And as you might expect, the primary drivers are conflict and insecurity, weather extremes, and economic shocks. These are topics that you will be addressing all week. We plant pathologists are observing an emergence of new pathogens and pests at alarming rates around the world. This graph shows just first reports, things that have been published, of crop pathogens globally since 2015 to 2022. As one example, In China, which is an important producer of many, many major crops, the relative area of cropland affected by damaging pests and diseases has quadrupled in the past half century and is easily expected to double again by the 21st century. Another recent report, a global analysis of potential changes in plant pathogens and infection rates with climate change, suggests that although in some areas we'll expect to see increased yields in crops due to the warming climate, the problem is at the same time we see increased diseases, risks for diseases. And this will, in fact, counteract any increase that we have in yield. So given the diversity of expertise and the brain power in this room, I'm confident that this week we're going to hear some wonderful ideas and brilliant ideas about approaches and schemes for how we might mitigate these problems in the near future. So I'd like to finish this morning by giving you a short story, so bear with me. Many years ago, I was on a flight returning from a meeting in the Philippines, and I was seated next to a doctor from the World Health Organization, or WHO. Now, this doctor was a chatty guy, and he spent probably several hours, talking about what WHO, the World Health Organization, had done in terms of introducing at that time, so this is in the late 1980s, oral cholera vaccines, how they were working towards reducing malaria globally, and how they had launched just in 1988 a global polio eradication initiative, which was the largest initiative globally that had ever been initiated, and in the history of public health. So as we were sitting there, he pointed out many times how many lives worldwide the activities of WHO had affected and had saved. So I was, 30-some years old, feeling a little bit insecure and thinking, wow, these doctors from WHO, these people are rock stars. And I was feeling pretty insignificant about that point. But he turned to me then, finally, and he said, so, what do you do? Well, I was a little bit quiet for a while. I was thinking, because I was kind of in awe of everything he had told me. And finally, I responded. I don't know where this came from, but I responded, I'm an agricultural scientist who studies plant diseases. We feed the people that you keep alive. We not only feed the people that you keep alive, but we feed, clothe, and house the people that you keep alive. So why am I telling you this story? So what I wanted to point out is that this was an aha moment for me. I was out of graduate school in my first faculty position for a couple of years, and this was the first time I really comprehended how important plant pathology and plant health specialists are to world health. And of course to human health, and to animal health, and to environmental health. So this conversation with the doctor from WHO generated or gelled for me just how important plant pathology is to One Health, the concept that we'll be talking about this week. And in other words, that you, the plant health specialists, are every bit the rock stars as the doctors from the World Health Organization. So thank you, and I hope you enjoy the Congress. So this Congress wouldn't have been possible without the efforts of our hosts, the French Society of Plant Pathology. So it's my great pleasure to introduce to you the president of the French Society, Sylvie GERMAN-RETANA, who will introduce our meeting and our meeting launch speaker. Sylvie is with INRAE and is the Deputy Director of UMR-BFP, which is a partnership between INRAE's divisions of biology and plant breeding and plant health and environment and the University of Bordeaux. Sylvie?
Thank you very much, Jan. So, bonjour à toutes et à tous. Good morning, everybody. So, on behalf of the French Phytopathological Society, SFP, it's really a great honor and also a real pleasure for me to welcome you all upon the opening of this 12th ICPP meeting in this beautiful city of Lyon. And first of all, I would like to extend my really warmest thanks to Mathias and Nathalie and all your colleagues and your committed team for making this Congress possible because Mathias and Nathalie have always been very, very, I would say, loyal SFP members. And I guess that organizing such a huge meeting is a huge amount of work, a real challenge, and a really big personal investment. So thanks again to both of you and all your colleagues I cannot cite who helped you in the adventure. And above all, try to have fun this week as much as possible, as much as you can. So yes, you can already. And I'm also very impressed by the number of people in this audience. So there is no doubt that the week's scientific and informal exchanges will be very interesting and exciting. And so thank you all for coming in such numbers. And as president of the SFP, I am also very, very happy and delighted that I will be able to meet many SFP members in real life, because about 1/3 of the SFP members are attending this meeting this year. So see you everybody soon. So now I was supposed to introduce the president of this meeting, Valérie Verdier, who appears on the picture. So Valérie is the official president of this meeting, but unfortunately, Valérie had a cycling accident a few weeks ago. She's doing fine, no worry, but she couldn't attend the meeting, and she will be immobilized for several years. But, so I'm going to introduce her. So I'm going to put my glasses on. So Valérie Verdier is the chairwoman of the board and chief executive officer of the French National Research Institute for Sustainable Development. French people is IRD. And at the time of her appointment, about 3 years ago, Valérie Verdier was working as director of the Plant Pathology Research Unit in Montpellier and headed up one of the 5 IRD scientific departments. So she contributed to the scientific strategic positioning of the IRD scientific policy in favor of sustainable development and fair partnerships with researchers in tropical and subtropical countries. As a plant pathologist, Valérie has been studying the interactions between phytopathogenic bacteria and tropical plants, an understanding which is really crucial to food safety for a large part of the world human populations. And she has co-authored over 100 scientific publications, and she is world-renowned for fundamental contributions to understanding the mechanisms by which bacteria cause diseases, especially those afflicting cassava and rice, and to the approaches developed to control them. So Valérie completed various short and extended international missions all over the world and in the course of her career. And in particular, she received a Marie Curie Fellowship from the European Commission in 2010, and she worked with Jans in Colorado State University. She received some awards, so the International Service Award from the American Society, Phytopathology Society, APS, in 2014, and she was also appointed to the rank of Knight of the French Légion d'honneur in 2016. And perhaps more importantly, Valérie really worked a lot as a coordinator of a program to help young researchers. And also she was deeply committed to train young researchers in developing countries, and especially women. So of course we miss Valérie today, and we wish her a speedy recovery. And so this is not Valérie who is going to talk now, but Bart THOMMA, who is the vice president of this ICPP meeting. And Bart really, really kindly accepted to give this introduction topic in the place of Valérie. So Bart THOMMA is known for his work with the vascular wilt pathogen Verticillium dahliae. And since 2020, Bart has held a Humboldt Fellowship in evolutionary and in microbiology at the University of Cologne in Germany, and at the Center of Excellence on Plant Science. And Bart received the first RKS Wood Prize, which is awarded by the British Society for Plant Pathology in 2018, oh, yeah, more or less, sorry. So Bart, as I said before, is the vice president for this meeting, and so I'm very happy that we can now welcome Bart to open the Congress. So thanks a lot for your attention. Have fun in this meeting, and I wish you the best for the rest of the week. Thank you very much.
Okay, well, wow. This is a very nice hall, very big, also a bit intimidating, I have to say. Thanks very much for the very kind introduction. And indeed, it is not me who should be standing here. It should have been Valérie. I'm not the only vice president, I should say. I'm one of the 4 vice presidents of this meeting, but when I got this request to step in, I realized, of course, I need to do this. And I guessed, well, Valérie had a biking accident, and of course I wish her a very speedy recovery, but I thought, well, maybe this is the reason why they thought you should ask, you should not take any risks with bikes anymore, so ask a Dutch guy to open this meeting. So let me start by saying that I think that our host, the French Phytopathological Society, and also especially with support from the scientific committee headed by DJ Tarot, put together an exciting program for all of you. And they brought it together under an exciting and challenging theme for the Congress, namely One Health for all plants, crops, and trees. I think you will all agree with me that this topic is extremely relevant. The topic is timely. I guess we can also say it's very urgent. But on top of this, I think it is also very appropriate. It is very appropriate for this ICPP meeting held here in Lyon today. By now, this One Health concept is worldwide recognized. It's a sort of strategy and for integrating interdisciplinary collaboration, communication, action plans covering all aspects of healthcare for humans, for animals, and for the environment. And here you see a typical logo. If you just Google One Health, you will see something like this. A logo that depicts this One Health concept. And you can already see that plants have their place in this logo. So that's, of course, very nice and very well appreciated by all of us. But then, if you look carefully, you see that plants also have their place, but as a representative of the environment. And of course, that's not completely justified. Plants deserve more attention, plants are more important, and particularly plant health and plant pathology are more important than appreciated here in this One Health logo. But a minute ago, I was saying that the topic is very appropriate for this meeting to be held in Lyon. So why did I now say this? For that, I'm going to introduce you to someone. So here you see, I would say, a fine young man. The hairdo and clothes tell us that we go back in time. So we go back in time, but not back in place. We end up somewhere around half of the 18th century, but we stay in Lyon. And so this is a representation of a person named Claude Bourgelat. He was born in Lyon in 1712. He grew up in Lyon. He studied in Lyon. He studied economics, and he studied law, and then started working as a barrister. And as a hobby, he liked to ride horses. And apparently, he had so much passion for riding horses that he developed his own riding style that I cannot demonstrate you. I'm not an expert at this at all, but apparently it's a riding style that's still being practiced today. So he was quite influential. And he was also influential in other ways, because in 1740, when he was only 28, he became the head of the Lyon Academy of Horsemanship. Roughly 10 years later, he authored a book on the topic of veterinary medicine. And because at that time, horses were such important animals, this book on veterinary medicine was mainly about horse medicine. At that time, Bourgelat saw that a disease, was killing their herds of horses. He got intrigued, and he felt the responsibility to start doing something. And that's where he discovered that actually he was much more a scientist than he was a barrister. So he decided to devote all his time to finding a cure for this particular disease, the disease that we nowadays know as cattle pest or rinder disease. Bourgelat started to align aspects of human and animal medicine, and that was very, very revolutionary at the time. He started something that he then called comparative medicine, initiating comparative pathology. And visionary as he was, he then founded the first veterinary college here in Lyon. Paradoxically, the separation of veterinary medicine from human health benefited One Health, the theme we talk about today, as it resulted in a flow of ideas from human pathology to animal medicine and vice versa. Really, a new concept of health emerged, leading to investigations of herd animal health, with findings informing human health. And while having come recently out of a pandemic ourselves, I guess for us it's now sort of easy to understand how important understanding concepts of herd immunity is, and of herd health, of course. And so I think we can say that this understanding really started here in Lyon with this fine young man, Claude Bourgelat, the founding father of the One Health concept. Today, One Health is typically defined as an integrated, unifying approach that aims to sustainably balance and optimize the health of people, the health of animals, and the health of ecosystems. And it recognizes that the health of humans, domestic and wild animals, plants, and the wider environment are very closely linked and depend on each other. However, in practice, the concept of One Health is mostly seen in the context of the control of zoonoses. with the discussion about the origin of COVID-19 and the avian influenza perhaps as the most recent and obvious examples of this. Typically, plant health and environmental concerns receive much less attention. Within the One Health framework, as it stands now, plants have no real independent own place as organisms with their own health problems, their own health concerns. But even if we only see plants as a source of food and feed for humans and animals, they deserve much more attention. They deserve better recognition, and I think they deserve their own place in the One Health concept. The first, and I guess most obvious reason, is that because of food production losses to plant pathogens and plant diseases that regularly leads to famine and malnutrition, as just explained by Jan, this is the first argument why plants deserve a more prominent place in this One Health concept. Another reason is because of the acute or chronic poisoning of consumers, both humans and animals, by toxins produced by pathogens in diseased plants. Yet another reason could be that crop health often requires the use of plant protection products, and that the often much more than desired, I would say, with negative effects on consumer health, either through the exposure to these products or through the consumption of their residues in food and feed. And another problem associated with these plant protection products is obviously the induction of resistance, also in microbes that reside in the environment, and that may again impact human health. A final reason, I think, why plants deserve a more prominent place in this One Health concept is because plant protection practices affect ecosystem services that are really important for our well-being. Thus, I think it's important to recognize that plants are organisms with their own health concerns and deserve more attention in this One Health concept. And with the current climate change concerns, the impact on plant health, this only gets more and more important. Thus, to conclude, I hope you can all agree that the choice of the One Health topic here in Lyon is not only extremely appropriate, but also very timely. And we should thank the organizers for integrating this concept here in the conference. With this, I'm going to close my speech and thank you all for your attention. I wish you lots of nice discussions. I wish you lots of inspiration, and I wish you lots of excitement, and hope you will all have a wonderful week here. And perhaps after that week, when you go back to your home institutes or your home countries, perhaps flying back or going by train, I hope you think back for a second to this fine young Frenchman who now in the 18th century basically laid the foundation for the wonderful week that we are going to have here all together. And with this, I declare that the conference ICPP 2023 officially opened. Thank you very much for your attention.
Thank you all, and thank you, Bart, for that inspiring opening. We're now turning to the part of the program for the Jakob Eriksson Award. This is the centennial year of the prize, the Jakob Eriksson Prize for Plant Pathology. It was established, as I mentioned, in 1923 at an international conference of phytopathology and economic entomology in the Netherlands. The prize is given to encourage the creative study of plant pathogens and the processes of disease development in plants. It was first awarded in 1930 to Professor John Hubert Craigie of the University of Manitoba in Canada. A fund established in the 1930s with contributions from 3 prominent international plant pathologists and the American Phytopathological Society has been administered by the Royal Swedish Academy of Sciences since the 1950s. So the Royal Swedish Academy of Sciences is the organization that issues the Nobel Prize. The funds that were donated are used to mint the Jakob Eriksson Gold Medal. So again, this is the 100th year of the Jakob Eriksson Prize, and it's named in honor, of course, of Jakob Eriksson, who was a prominent Swedish mycologist and plant pathologist who wrote prolifically in 4 languages and promoted cooperation between plant pathologists or among plant pathologists. Eriksson's writings and teachings and ideas were instrumental in the formation of the International Society of Plant Pathology in 1968 at the First International Congress of Plant Pathology. So to award the Jakob Eriksson Prize, I would like to welcome Dr. Dan FUNCK-JENSEN, Chair of the Jakob Eriksson Commission, to the platform.
Good morning. Can I call Sylvia Restrepo to the stage? So, on advice of the Jakob Eriksson Commission, with endorsement by the International Society for Plant Pathology, the 13th Jakob Eriksson Prize is awarded to Professor Silvia Restrepo of the Universidad de los Andes, Bogota, Colombia. Professor Silvia Restrepo is being recognized for her pioneering international work in mycology and plant pathology with a focus on diseases that impact crops important to the developing world, such as cassava and potatoes. So I have the gold medal here for you.
So it's my pleasure now to invite Professor Silvia Restrepo to deliver the 2023 Centennial Jakob Eriksson Oration.
Thank you. Thank you so much, and it's indeed intimidating to be here in front of you. I would like to thank the Swedish Academy of Sciences and the organizer. Merci beaucoup, Nathalie, Mathias, et tous les organisateurs. Merci. I have to switch between 3 languages. That's my life, between Spanish, French, and English. So I'm going to talk, and I chose to give you a glimpse of my research activities, and I call it my journey studying plant pathology in Colombia, more rewarding than challenging. And I would like to start thanking my mentors and thanking them the time and effort they have put into my career. Valérie, I was her first PhD student in her career. Jan, of course, and Joe Thome in Seattle, who was my mentor, Chris Mart and Bill Fry at Cornell University. And I chose to talk about two pathosystems, and the first one was the one I studied during my PhD studies. It was the interaction between cassava and Xanthomonas phaseoli pathovarmanihotis. Us plant pathologists like to change the names of the pathogens. When I started, it was not called Xanthomonas phaseoli. It had another name, but anyway, it was the interaction between this pathosystem I started my career in a disease that is caused by this bacteria, that is the cassava bacterial blight, CBB. And I have always chosen to work with the small growers. So you can see some of the pictures we took at the time. I'm talking about the 90s, late 90s I did my work. So during my work with the small cassava growers, I always start with the basics and in Colombia with the population genetics to understand the diversity and how this diversity distributes in their geography. We studied this population genetics with traditional markers, so the younger people in the room don't understand what these pictures are, but RFLPs, AFLP, the traditional markers we used we use in the time. And we studied the diversity of the pathogen in three developing countries, Colombia, Venezuela, and Brazil. This was the work I did with Valerie. At the time, we found significant difference in haplotype and pathotype frequencies over the years, showing that the population of the bacteria are very unstable. And this is important because it has consequences on when and how to monitor the dynamics of these populations. And we also understood that migration shaped the population structure of the bacteria, and this again determines how to select the cultivars to be deployed in each of the ecozones we identify in the three countries. The most important result of this research was that we worked with the breeders and we selected cultivars for each ecozone to improve the crop's resistance to CBB. We did this work in the field with 19 cassava cultivars, and we selected some resistance. This work was done in the International Center for Tropical Agriculture in Cali, Colombia. But then most part of my career was focused in the study of the interaction between solanaceous crops and different species of Phytophthora. And here again, some of the pictures of the plants, I'm gonna talk a little bit more. But actually, I started this at Cornell when I was a postdoc in Chris Mart and Bill Fry's lab. And we started trying to understand the molecular interactions between the pathogen and the host in a compatible interaction. So we found in one study that this enzyme, the carbonic anhydrase, had an important role in the compatibility of in the compatible interaction. Actually, when we silence this enzyme, you see here, when we measure the quantity of the pathogen, we quantify the pathogen in the plant, that when you silence the carbonic anhydrase, the pathogen can grow more quickly, indicating that this silencing, the suppression of this enzyme increases the susceptibility to Phytophthora infestans. And an important and interesting result that we showed in these studies is the largest group of genes that were suppressed during the compatible interactions were the photosynthesis-related genes. Years later, in my lab, we studied again this interesting suppression of the photosynthesis, but with a different approach. And we used the metabolic reconstruction of the plant during compatibility to understand how the photosynthesis is shut down. And so we reconstruct the metabolism of the plant during the interaction. And you can see here how a long time during the interaction, the compatible interaction, the metabolism of the plant changes dramatically. And you can see that at 6 hours, all the metabolism is totally different. And all the dots are the metabolites and the lines are the metabolic pathways. And we found that the key or pathway of this shutdown of photosynthesis during the compatible interaction was the synthesis of the glutamate. And so we did this, and I invite you to see this paper because it has interesting results on how the photosynthesis is shut down. But then I went to Colombia again. This is 2005, and I started to go to the field whenever it was possible. You know that Colombia suffered a long time of violence, and we couldn't go to the field, but I used to go sometimes to the field. And doing field work in Colombia is really challenging. You have here a potato field that starts to the right, but in the garden of the grower, he has at least 4 different hosts of Phytophthora infestans. So you have Physalis peruviana, you have Solanum quitoense, you have pepper, you have tree tomato. So it's interesting because he keeps the reservoir of the pathogens for his field. So it's really interesting. So when I started my line of research, or my lab in Los Andes University in 2005, I started several lines of research. First, we wanted to understand what were the hosts of Phytophthora infestans. Of course, we monitor the populations of the pathogen, and we keep studying them until today. We did some genomics works. We established the pathogenicity and virulence determinants, and we try to find alternatives for Phytophthora infestans control, if that's possible. Everybody who works with Phytophthora knows that it's an interesting question. So I'm going to go through all these lines very quickly. And first, as I mentioned, we established the host of the pathogen in Colombia. And these are on some of them that we could establish and do the Koch postulates. Of course, the traditional host, potato, tomato, but also all the solanaceous crops you find in the Andes. We are the center of origin and domestication of all these crops. So we have the tree tomato, Solanum betaseum, the lulo, Solanum quitoense, uchuva, or Cape gooseberry, Physalis peruviana, and a wild species, Solanum viarum. And knowing all these different reservoirs, we could go and monitor the populations. The first study we did in the population, or studying the populations, the population genomics or genetics of this pathogen was in 2008. With traditional markers, again, RFLPs, the traditional markers that are used in the Phytophthora research to establish the clonal lineages. And we found that a new population was established in Colombia, replacing the traditional population that was described in the 80s and the 90s. And I found a very interesting result. Only one strain that has the A2 mating type. You know, Phytophthora has 2 mating types, A1 and A2. And I found in this collection, the first collection, one strain that had the mating type A2. All the other strains in Colombia show the A1 mating type. And among more than 1,000 strains that have been characterized in my lab, this was the only one that was characterized as an A2. And that's a mystery that still haunts me. How this mating type arrived to Colombia, we have tried to establish different hosts, and it's so an open question that we always have in science. Then we studied the population with different markers, and we use genes, nuclear, mitochondrial, and some virulence genes to study the populations. 2 important things of this study. You see here Colombia, you see Venezuela. We studied the populations of Phytophthora infestans also in Venezuela. And the first important result is you can see we found different haplotypes in Venezuela, in Colombia, depending on the marker. And the second one is you can see here in the south of Colombia this huge level of diversity. I'm going to come back to this story later. And then we continue I try to monitor the populations to see how they change and help the breeders every 2-3 years. And this is the last one we did in 2019. And we try to see in this study whether the geographic origin or the potato genotypes shape the population structure of Phytophthora infestans, and we couldn't establish a relation. You can see here the haplotypes, and you can see that the same haplotype can be collected in a different location of the country, or the same haplotype come in different genotypes of potato. So neither the genotypes nor the geographic origin shaped the population studies. And this was done with the most important breeder of potato in Colombia, and just a couple of weeks ago he released 3 varieties of potato that result from the collaboration with my lab. Jackie, Sipa, and Villa are the names of the 3 potato varieties he released a couple of weeks ago. We also monitor the resistance or sensibility to fungicides. And it's important we established the baseline a few years ago, and again we took the picture in 2019, and the situation is the same. We see that the strains have a lot of resistance to the traditional mefenoxam, and some other molecules still can control the disease. And we also studied the population in tomato with some collaborators in AgroSavia, and you can see something that is that we see all the time. All the strains from different hosts. In dark blue, you see the ones collected from tomato, and in light blue, you see the ones from potato. In yellow, the one from tree tomato. And in green, the ones from the US, the reference strains. And you see that they always separate genetically, and they have a strong host preference. As Berg showed a century ago, we also showed that the strains from tomato have a really strong host preference. These are strains isolated from potato and inoculated in potato and tomato, and you see that the potato strains cause more disease in potato host, and when you collect tomato strains, they can infect tomato, and very hardly they can infect potato. So we see a very host preference. And we also studied the resistance and sensibility of these strains from tomato. In the top 2 rows, you see the strains from the US that we use as a reference, and then the bottom 2 lines, you see the ones collected in Colombia. And you can see that still we have this resistance to a lot of fungicides or oomycetes, because it's not a fungi, it's not a fungus. But anyway, it has a lot of resistance. Still, again, this same molecule, flupicolide, has some, shows some the strains show sensitivity to this molecule. But we are seeing here an important result is like the resistance to all these products that we shouldn't be using. But anyway, so you remember the story about the huge level of diversity in the south of Colombia, close to Ecuador. Ecuador is here, Peru is here, and so we try to understand what was happening in the southern part of Colombia. And going back to the literature, I found this paper by my colleague and friend Maria Eugenia, who showed that the novel population of Phytophthora, similar to Phytophthora infestans, was attacking wild Solanum species in Ecuador. And also my friend Ricardo Oliva and collaborators showed and called this new population Phytophthora andina. But these species had several clonal lineages, and that was something that I tried to understand a little bit more. It grouped strains with a clonal lineage EC2, EC3, and EC2 and EC3. So going deeper in this to understand what was happening with this species that probably was phylogenetic, we described actually that there were 2 species in this Phytophthora andina species that Ricardo described. And we found that actually we had 2 species, Phytophthora andina, that corresponded to the clonal lineage 2, and a new species we described with my students and colleagues that was the EC3 lineage. And this species shows a very host preference again, and we can see that in the field we cannot find this species, Phytophthora betaceae, in potato or tomato. We have described only in Solanum betaceum, the tree tomato that has big trees. So we have here a case of ecological speciation. The 3 species are monophyletic, and the most probable scenario is a speciation with secondary contact. And we also showed that they have biological separation. We couldn't see when we did the crosses between infestants and the new species. We don't see viable oospores, so the hybrids are absent. We have a reproductive isolation. So indeed, we have 2 separate species. And these species coexist in the northern and southern part of the country, and you can see in green the ones isolated from potato, that is orange, the ones isolated from tree tomato, and they coexist in both parts of the country. And you can see here that actually they separate between the P. andina, P. betaceae, P. infestans, and regardless of their origin, they cluster together. So we look into the genome of these new species, and we found that this genome has a whole genome duplication, is huge, is bigger than the one in P. infestans. Here we compare the genome of P. betaceae and the P. infestans one. And you can see that most of the genome comes from segmental duplications. And in this picture, you can see better how segments of the genome of the Phytophthora infestans are duplicated in the genome of Phytophthora betaceae. And when we see how the effectors and the virulence genes show in the genome, you can see that we identify the ones in P. infestans, the one in P. betaceae, the ones that are unique to P. infestans, the ones that are unique to P. betaceae, and the ones that are shared. And you can see that always, if we speak of the apoplastic effectors, of the crinkles, of the RXRL, P. betaceae has more effectors than the P. infestans alone. And the important question also is we are studying these ones that are unique to P. betaceae, try to silence them, but as you can imagine, they are duplicated, and it's really hard to silence these genes. So, and to finish, we also try to find alternative for control, and this is an interesting story. We chose the pyrimidine biosynthesis pathway, and in this organism, you have 2 pathways to synthesize pyrimidines, the de novo synthesis pathway and the salvage pathway. And why this pathway? Because some of these enzymes that are responsible for the synthesis have been attractive for drug development. And we found, first, that is really interesting, the de novo enzymes are upregulated during the biotrophic stages of the cycle, you can see here, and then the salvage enzymes are downregulated during the de novo pathway, the biotrophic stages, and then upregulated during the necrotrophic stages of the disease. And in the literature, you also find that this enzyme in humans, the 4 enzyme, here that is the 4 enzyme of the de novo synthesis pathway, has been attractive for drug development. And remember, this name is complicated, but this compound, remember the name. And another compound has been used for the apicomplexan parasite Plasmodium falciparum, and is currently undergoing, was currently undergoing phase 2 clinical trials in 2017. And remember that the apicomplexa are very close phylogenetically to the mycota and Phytophthora infestans. So that's why we chose these products. So with a student and a collaboration with Howard Judelson in California, we studied the same, well, we studied the fourth enzyme in Phytophthora infestans, and we showed that it has a mitochondrial localization, and we used the compound that I told you to remember, the one that is used against the human enzyme, and it inactivates the pathogen. It can control. Here we compare with a fungicide, and you can see that at higher doses, it causes low growth of the pathogen. And an important consideration, it does the solanum enzyme, the potato enzyme, and the Phytophthora enzyme have different sensitivities to this compound because we don't want to inactivate the enzyme in potato. So in conclusion, studying plant pathology in Colombia, and in Colombia, agriculture is a very important sector of the economy that has 16% of employment and 7.5% contribution to the gross domestic product, with significant challenges. Low productivity, is not data-driven, it has low adoption rates of technology. We don't have impartial extension services in Colombia. Extension services are done by the big companies, and only 0.2% of the GDP is invested in research. So that's how I highlight the importance of continuously monitoring the pathogens. Us plant pathologists have this mission to monitor the populations with the appropriate markers that allow discriminating variants at a local and a global scale and under particular environmental conditions. And the importance of working with breeders to select the pathogens variants to use in selecting resistant sources. We need to understand the relationship between the genetic structure of the pathogens and the nature and distribution of pathogenicity and virulence. An important legacy of my work, and everybody in this room probably, is to mentor young scientists, especially in Colombia, to open the world for them. And these young scientists are currently senior scientists or professors in the best universities, Stanford, Max Planck, Hôpital de Genève, MSU, and some of them are here today. So please raise your hand if you were in my lab, as a student, as a collaborator. So, thank you. Come on. I know that, yeah, some Colombians here. And please raise your hand if you mentor a student that came from my lab. Thank you, Jan. So, two final messages. If the conditions change, we can and we must help the most vulnerable. We have the knowledge, the technology, and the vocation. When the pandemic came to our life, I changed the whole university, and I opened the labs to help running PCR diagnostic tests. We run 170,000. We help applying 110,000 vaccines in 3 poor neighborhoods of Bogota, and we sequence all the genomes that were circulating in the country. And also, we have to contribute to attain the Sustainable Development Goals and the sustainable use of biodiversity. These are some pictures that from my work. I didn't have the time to show all my work today, but I work with Cordyceps amanita. Every time I go to the field in Colombia, we can describe 1 or 2 new species for science. So we have this huge diversity that we need to understand and use in a sustainable way. So I would like to thank my collaborators all over the world, and my beautiful family, my husband, who is here, my brother, my friends. My son couldn't come because he's at the university taking classes. And this is my new challenge. I will be the next president of the Boyce Thompson Institute, and I would like to invite you to collaborate with us now at BTI. Merci beaucoup. Gracias. Thank you.
Thank you, Sylvia, for that inspiring talk. I think you can understand why she was awarded the Jakob Eriksson Prize. I'm reminded of a story that you'll now understand. When I was editor of annual reviews of phytopathology, Sylvia was invited to write a review. And of course, she turned it in beautifully on time. Everything was great, but it was twice as long as they're supposed to be. And so I wrote to Sylvia and I said, well, Sylvia, we're only allowing so many pages. I think it was like 16 or 20. And so we're going to have to cut this. And she said, well, I tell you what, why don't we just split it into 2? We didn't. But that was a telling sign. And I also want to point out she did it while she was dean. Now she's much higher in the ranks. But I can't believe anybody could do that much while they were dean. So we're now at the point in the program where we need to not only recognize the dynamic duo who have worked so hard to pull this meeting together, but also to let them tell you some logistics about the meeting and tell you what's happening. So it's my great pleasure to introduce my now best friends in the world, Doctors Nathalie POUSSEREAU and Mathias CHOQUER.
Thank you, Jane. Dear ladies, dear gentlemen, dear colleagues, dear friends, welcome in Lyon. Bienvenue à Lyon. The local organizing committee, our professional congress organizers, Europa Group, and all the volunteers are very, very happy to welcome you in this beautiful amphitheater. We are very, very proud to organize the 12th International Congress of Plant Pathology. But you know, the organization of this meeting of ICPP 2023 was a big challenge for us. Why? Because we had to overcome several difficulties. And several times we wondered whether this meeting would take place or not. We had to overcome the COVID crisis, as you know, and several social events here in France. And finally, because of international conflicts and war, several colleagues cannot join us today. So that's why a concurrent session focusing on impact of war and conflict in plant pathology research will take place this afternoon, and we invite you to participate. Despite all these events, we are finally more than 2,400 attendees coming from 100 countries. So it's not so bad. The topic of the meeting is also challenging, as previously discussed by my colleagues, how plant health and plant pathology can be introduced in the One Health concept. A big question. And we hope, and we are sure, that this meeting will give you the opportunity to have successful discussions. To facilitate this, we have developed interactivity with different tools and events that Mathias will present you in a few minutes. Of course, we want to thank the scientific committee and more particularly Dr. Didier Tarot for his great help in the organization of this meeting. Yes, I think that we can see. Thank you, Didier. I don't know where is he, but he's just over there. Yeah, thank you, Didier. Also, we want to thank all the colleagues who proposed 6 keynotes and 63 concurrent sessions. You could also participate to 9 very interesting roundtables. And finally, you will discover around 1,400 posters presented during 4 viewing sessions in the hall. Finally, we want also to thank our sponsors and exhibitors for their contribution to bursaries, and it was very important. We raised a bursary fund of around 150,000 euros to support 150 students and researchers coming from developing countries. A special thank to ISPP and to the French Institute IRD for their important contribution to bursaries. Mathias?
To continue on the sponsors, yes, we can applaud the sponsors. Thank you. To continue on the sponsors, we will invite you to visit their booths, the booths of the exhibitors, who have 20 booths in the exhibition. And so you have the exhibition plan in the paper program at page 127, and you can also see the map of industry partners in the app. Maybe you already know that we have a Wi-Fi connection, so this is the network's name of the Wi-Fi and the password. Don't forget to download our app to get social, and eventually you may ask away your questions to speakers through the app. You will have to open the app, choose the interactivity feature, and then choose the room you are in. So chairs are supposed to read your questions and to select some of them. We have also other tools for interactivity. We would like to thank Diana Fernandez to organize this event, that if you want to talk to an expert, you can organize your own meeting with one of our 40 experts. This will be organized during a poster viewing session. So we'll be waiting you near the poster area in number 3 to sign up for a 15-minute slot, and you will be able to write your name on a special board in the one-to-one conversations with an expert area. So please note that places are quite limited, so they will be located on a first-come, first-served basis, and only one slot will be possible with one expert to be reserved. We'll have also another tool for interactivity. We would like to thank Marc-Henri Lebrun to organize this. It will be discussions with keynote speakers during lunchtime. So you may sign up by sending a message directly to the keynote speaker using the ICPP app. And the meetings will take place in a specific spot near poster area 3. You will have to bring your lunch from the catering of the Congress. Keynote speakers from Keynote 1, so you can meet them on Monday. For Tuesday, it will be keynote speakers from Keynote 2 and 3, Wednesday, Keynote 4 and 5, and Thursday, Keynote 6. As Nathalie mentioned, we have several roundtables, so we would like to thank all organizers of these roundtables that would help to have more interactive discussions. On Monday, you will have a roundtable on getting rights right. It will be an exploration of indigenous rights and participation in plant pathology. And a second one in management of post-harvest diseases in Mediterranean countries to reduce food waste. On Tuesday, a roundtable for a discussion and forum on cross-cutting issues generated by findings of the Global Plant Health Assessment, and a second on the pathobiome, new understanding of post-harvest diseases, and a third one, two roundtables on Tuesday. On Wednesday, we'll have 3 roundtables, one about the impact of discoveries in plant health, the second on sharing and exploiting high-throughput sequencing data, HTS, and the third one on germplasm seed movement and global plant health. And finally, on Thursday, we'll have a talk during a roundtable on research integrity in plant pathology, and also we will open a discussion for PhD students and postdocs with our industry partners. It will be about jobs in plant pathology and how a plant pathologist works in industry setting. So all these roundtables will take place during poster viewing sessions. So I want to thank all organizers again from these roundtables. We would have some spatial temporal recommendation, especially as we are a lot of people. Places will be quite limited in the concurrent session rooms, so we would ask you please to not leave an empty seat between you and your neighbor. Please occupy all seats to prevent people from standing. And we also thank speakers for not exceeding the time allocated for their oral presentation, and the chairs for being the timekeepers. Finally, speakers, they must upload their PowerPoint presentation as a preview in the Belcourt office, in the level Belcourt, the day before their speech. Please note that on Friday, the final day of the ICPP Congress, we will have to move to another part of the Congress Center building. We'll have to move to the Terreau entrance. So the people that were at the workshop during the weekend, they already know the place. So be careful, because on Fridays, the Belcourt entrance to the amphitheater will be closed. We will also have the Only Lyon Convention Bureau. And so this desk will be staffed with a Lyon expert who will answer questions and assist you with diner reservations, tours, and activities in Lyon. They will be present for 3 days, Monday, Tuesday, and Wednesday, and the place is in the Hall Belcourt, in the welcome area. As you know, we are experiencing a strong heat wave, so how to stay hydrated? You can use several Congress water stations and drink water throughout the Congress with our ICPP cup. So I think we finished this presentation, so we would like you to enjoy your congress. And we will start the first keynote session at 10:30 in the amphitheater. It will be about plant pathology in a one else word, chaired by Brenda Wingfield from South Africa and Yvan Sage from France. And now we propose you to socialize and have a coffee break of 30 minutes in the forum, and we thank you all.
Thank you.
Thank you.